So good morning. Hello, and thank you for joining our Introduction to Statistics webinar today. I'm Polly Kerr, and I'm the Patient and Public Involvement Manager for the Oxford Biomedical Research Centre. And together with my colleagues, Claire Murray from the Oxford Health Biomedical Research Centre, Claire Schwartz from the Applied Research Collaboration for Oxford and Thames Valley, and three of our public contributors, Hubert Allen, Kath Greehey and Julia Hamer-Hunt. We are hosting these webinars for patients and members of the public who are involved in research or who are interested in getting involved. In a minute, I'll introduce our speaker, but as I've said, we're recording today's session. That's so that we can make it available on our website afterwards. So please keep your cameras off and your microphones on mute, particularly because we are expecting quite a large number of people today. I think there's still people joining. In a minute, Jackie's going to speak for about 45 minutes, and there'll be a sort of 15-20 minutes time for questions after the talk. Please do submit these using the chat function while Jackie's doing her presentation. And just a reminder that these webinars are aimed at increasing patients and the public's knowledge and understanding of research, so the questions will be prioritised to reflect that aim. Finally, from me, we'd love your thoughts on today's webinar. So we'll be sending you a very short questionnaire via email later today, and we'd be really grateful if you could fill that in, because we'll use all the feedback for planning for our future webinars. And now I'm really pleased to hand over to our speaker for today, Jackie Birks, who's a senior statistician with the Oxford Biomedical Research Centre. Thanks, Jackie. Thank you for being here today.
Can you all see that? Yes.
Perfect.
Right, brilliant. Thank you. Well, thank you, Polly. Thank you for inviting me, and thank you all for joining us today. I can see that you're joining from not just all over the country, but from internationally as well. I'm Jacqueline Birks, and I'm the Senior Statistician for the NIHR Oxford Biomedical Research Centre. I'm also a research advisor for the Research Design Service South Central. The Research Design Service provides advice and support for researchers applying to the NIHR research programs, and also other research programs such as those run by charities. Well, it's a great privilege and a pleasure to be able to talk to you today about medical statistics, medical statisticians, and their role in medical research, one of my favourite subjects. Well, clinical research is multidisciplinary. It cannot happen without collaboration, and medical statisticians are a part of the clinical study team. As PPI contributors, you are experts in many fields and disciplines with greater or lesser knowledge about statistics. So where do I begin on an introductory talk? Some of you have sent questions to Polly and suggested topics that you'd like me to discuss, and I thank you for your ideas, and I have listed some of them here. So why are statistics presented with confidence limits and a p-value? What is risk? What is relative risk? What's a hazard ratio? Could you explain a forest plot? Kaplan-Meier plots, explain the results of the trial of dexamethasone for COVID-19. As you can see, technical questions have come up frequently, and there's a topical question about a COVID-19 trial. So this is all very interesting, quite challenging, and I shall have a go. But these are not issues that can be dealt with in great depth in 40 minutes. So where do I start? I'll start by telling you a little bit about statisticians. We are applied mathematicians, basically. Many of us have a degree in mathematics or a subject with a fair amount of mathematics. For example, I have a degree in physics, and this is often followed by a master's degree in applied statistics. And it's during that master's degree that we learn how to apply statistical methods. And there are many areas that use statistical methods, and therefore many opportunities for employment for statisticians. And I have listed a few here, not just medical research, agricultural research, marketing, sports statistics, very popular. Imagine watching your favorite sport all day as part of your work. Got to be able to do the technical stuff as well though. Marketing, econometrics, quality control in manufacturing, the pensions and insurance services, social sciences, all departments of government and financial services. The list is long and there's not enough statisticians to fill all the roles available. There is a serious shortage of statisticians. But I hope that the higher profile that medical statisticians have seen in this past year leads to an increase in interest in pursuing this career. I should like to tell you a bit about the RSS, the Royal Statistical Society as well. The Royal Statistical Society is the nearest we have to a professional body. It does not regulate statisticians though, in the same way as, for example, doctors and lawyers are regulated by their professional bodies. And the Royal Statistical Society have kindly provided me with slides which they use for promoting their society. So they're a charity which promotes statistics, data, and evidence for the public good. They're one of the world's leading learned societies and the only UK professional body for all statisticians, by which they mean statisticians in all areas. And their vision is of a world with data at the heart of understanding and decision making. It's a very old society founded in 1834 and one of the world's leading organizations to promote the importance of statistics and data. It has a membership of about 11,000, of which 6,000 are designated as fellows. They're likely to be the statisticians who are actually actively employed as in a professional capacity. Note how small the profession is. I said there was a serious shortage. Not every statistician will join the RSS, but many do. The RSS is concerned with professional development and accreditation pathways. It also publishes peer-reviewed journals on statistics. It holds regular scientific meetings throughout the year. Well, it previously did. They're all online now, either in their headquarters in London or else in their local groups, and it includes an annual conference. They share their knowledge and expertise through volunteering. It runs a scheme whereby RSS members volunteer to help small charities with any work that needs a statistician, such as running a survey. They have this professional award scheme. And you may find that statisticians have a grad stat or C-stat after their degrees listed in their CVs. So these are accredited by the RSS, and it recognizes the combination of their experience and education and competence. And their awards are a measure of assurance to employers, contractors, and people like yourselves who have reason to talk to statisticians. Notice the data analyst coming soon. You've probably come across studies which use large data sets of routinely collected data, such as the hospital records, cancer registry data. Other methods, often called machine learning methods, are being used, as well as statistical methods, to analyze these data sets. If you look on the RSS website, you will find very helpful information on statistical issues associated with the COVID-19 pandemic. Next question, why do we need statistical analysis? because of natural variability. No people respond in the same way to a treatment or to a hazard that may cause disease. We have seen great variation this last year in the severity of illness caused by COVID-19 infections. The methods of statistical analysis are based on the fundamental idea that we use data from a sample to tell us about the population as a whole. I'll tell you a little bit more about sampling. The idea behind sampling is that the information gained from a single sample from a population is used as the best estimate for what is true of that population. But the sample estimate may be different to the true population value due to sampling error. It's really important that the sample is representative of the population in order for the results to hold any value. A well-chosen sample can yield reliable answers, whereas an unrepresentative sample produces misleading conclusions, often what we would call a biased sample. What causes these sources of variation? Well, let's think of something which we've all got experience of, blood pressure measurements. And we know that there's variation caused by lots of factors. Smoking causes it to increase blood pressure, diet, alcohol consumption, age, body mass index, and then there's unknown variation which is suspected but difficult to measure, like blood pressure often varies within a person with the time of day. And then there's other sources of variation, some of which are unsuspected and which are unmeasured. So what do we do about it? What do we do about random variation? Random variation between people in an outcome measure, for example, in one of the COVID trials, the outcome measure may be infection, whether they show infection, must be considered when designing studies with the aim of measuring a treatment effect. Now, the greater random variation there is within your population, the greater number of people you need in your sample. The way we deal with this is randomization. The design of the study can be used to limit the effect of variation between the participants by randomizing the participants to the different treatment arms. We're then more confident that the variation we observe is due to the treatment. We're making the assumption that then it's only the treatment that they're getting that's different between our two groups. Randomization's got a very interesting history. The randomized design was first introduced by R.A. Fisher, a Cambridge mathematician, genius, who laid the foundations for modern statistical science. Early in his career, at the beginning of the 20th century, he was working at Rothamsted Experimental Station, which is an agricultural research station in Hertfordshire. It still exists today. It's a brilliant place. He was analysing data from the field trials of crops. And there he established many of the techniques that we use today in statistics. In 1925, he published Statistical Methods for Research Workers, and it's still one of the most important books on statistical methods. The agricultural scientists were well aware of variation in the field. They knew that there were sunny spots, shady spots, wet spots, dry spots, different types of soil, different levels of fertility. And if they were testing, for example, varieties of winter wheat, they would divide the field up into areas that they considered more uniform. And then they would ensure each variety had a plot in each type of area. Well, Fisher formalized this approach with the 3 principles of experimental design: randomization, replication, and local control. The field was divided into blocks, which were then divided into plots. The varieties were randomized to a plot within a block, and this was the randomized design. And he then developed the theory to analyze data. These are the basis of the trials that we still use today in medical research, and they are based on the principles developed by Fisher. Well, today we are, of course, going to talk about clinical trials. And I have listed here 3 types of clinical trials, which you probably have met in your work. These are all design trials. We're not talking about observational studies today. That's another completely different and large area. Phase 1 studies are small trials investigating the safety in healthy volunteers. These are usually the first in human trials. They're not blinded, they're not randomized, and it's the first time you test a device or an intervention in humans. Phase 2 are larger studies, larger trials investigating safety in patients. They also can invest dosing levels of the intervention of the drug and the time between doses. They can be single arm, which means everybody gets the intervention, or they can be placebo controlled. They can be blinded or non-blinded. Phase 3 are the large trials investigating safety and efficacy in patients. They are usually double-blind, randomized, and controlled. These are the ideal trials. We can't always stick to them. Some trials are quite difficult to blind, like device trials and surgical trials. And just occasionally you'll find patients actually being tested in the phase 1 trial. The results from the phase 3 studies are required by the regulatory authorities such as MHRA, the Medicines and Health Products Regulatory Agency in the UK, and the FDA, which is the Food and Drug Administration in the US. And these agencies are responsible for ensuring that medicines and medical devices work and are acceptably safe. I'm now going to show you a simplified overview of a clinical study. First, we start by defining the research question, and then we have to set up a study design. And then to meet that study design, we have to write a protocol that describes absolutely everything that is going to happen in that study. And part of that protocol would be the detailed statistical analysis plan. When the protocol is ready, there can be an application for ethics approval. And after that, we can start the study, start recruiting the patients and consenting the patients. Data collection, of course, goes on throughout the study, beginning at baseline until the end date. The end of the study is defined as the date when the last patient to enter the study finishes their time in the study. After that, the database is locked by the medical statistician, and you can unblind the patients and start the data analysis. And of course, the final step is writing up the study report. The trial statistician will be involved in every step of that process. Now, I have mentioned right at the beginning and in the multidisciplinary nature of medical research. I'm now going to show you the front page for the Oxford AstraZeneca phase 3 vaccine trial, published in the Lancet Medical Journal in January of this year. Details of this study are frequently discussed in the news. In fact, recently they hardly ever seem to be out of the news. There's even a link on the BBC website to the paper, so it's very easy for anybody who wants to read the original paper to get a copy of it. Now, this is the front page. Notice the long list of authors. Those with an asterisk against the name are considered the main authors, and they all contributed equally. Now, if you look down the very bottom, you will recognize the name of Sarah Gilbert and Andrew Pollard from the Oxford vaccine group, who helped develop the vaccine. But do you recognize the name of the first person on that list, Merin Vosey? Well, Merin Vosey is the trial senior medical statistician. Let's go to look at the end of this paper and see their contributions. So at the very end of the paper, we have listed the contributions that the different authors made. And we can see that Merin Vosey is listed as contributing to the protocol and design of the study. She also did the statistical analysis, and she contributed with Andrew Pollard to the preparation of the report, and all authors critically reviewed it. So you can see, you begin to get some idea of the role that the medical statistician played in that Oxford AstraZeneca study. As I promised earlier, we were going to look at some of the trials of the COVID vaccine. And here, this is the same trial, the Oxford AstraZeneca COVID vaccine trial. And here I've extracted features of the design from the paper. It actually consisted of 4 randomized blinded controlled studies conducted in the UK, Brazil, and South Africa. The participants were aged 18 to 55 initially. They limited it to younger people, and later it's extended to people older than 55. Later, they also focused on healthcare workers. The treatment was the vaccine, the Oxford AstraZeneca vaccine, or the control group was a meningococcal vaccine. Now, that is a vaccine for meningitis, and there's not too much explanation in the paper as to why they chose that as the control. Well, I think the reason they chose it is because it's not likely to do anything against the COVID-19 virus, of course, but the vaccine for the COVID-19 virus is likely to cause some mild side effects, flu-like effects after you take it. You may get a headache, you may get a bit of a temperature. They're very transient, they don't last more than a day or two, but you may notice them. And so the trialists were worried that people would be able to guess which arm they were in if they had a saline control, which means it's placebo, it doesn't do anything at all. And so if they started getting those symptoms, they might guess that they'd had the vaccine, and that may have altered their behavior. They may have taken more risks, say, because they thought that they'd had the vaccine. And so I think that's why they used this meningitis vaccine as the control, which was likely also to cause some mild side effects. They did later move to using a saline control, which is an inactive control. The dosing was 2 standard doses, which are called SD. They were some days apart. Now that varied between 50 and 91 days. And there was quite a lot of discussion and talk and controversy about that. And the reason for the variation turned out to be that there were supply problems from the people who were making the vaccine. They were getting this trial up very, very quickly. from the actual making of the vaccine to the manufacturing of it and getting it to the phase 3 level. And there were supply problems. And also another problem was caused by some participants receiving a half-level dose, which they call low dose, as the first dose. And this was caused by a problem with another one of the manufacturers of the vaccine. And when it arrived in the UK, they were retested to check the concentration of these vaccines. And they discovered that those coming from a certain supplier were providing half the concentration that they had asked for. But by that time, they had already used it in some people as the first dose. So that explains the problem with this half dose as the first dose. It was suppliers' problems. Now, the primary outcome was infection with COVID-19, and it was confirmed by the NAAT, the NAT test, nucleic acid amplification test. That's the most accurate test we have. And it was confirmed from 14 days after the second dose. So let me just explain a little bit more about how we define infection. The participants reported specific symptoms associated with COVID-19. So they were asked to report these. This is following on 14 days from their second dose. And if they reported the symptoms, they were given a NAT test. But also, all participants provided weekly swab for a NAT test to identify those who may have been infected but were asymptomatic. Now, here we come along to the interesting part, which you have asked me about. I've got up here the results for the patients who had 2 standard doses. And you can see that we have about 4,500 randomized to each arm. We had 27 infections in those who had the vaccine, and 71 in the control arm, 0.6% infection in the vaccine group, 1.6% in the control arm. You divide the 0.6% by the 1.6 to get the relative risk of infection, which is 0.382. And the efficacy, and this is what you've heard reported many times in the news, efficacy of the vaccine trial is 1 minus that relative risk converted to a percentage by multiplying by 100. And that comes out to about 62%. What we're saying is there's about 1/3 of the risk in the vaccine group for getting infected compared with control. I've just added a note here to show the efficacy actually reported in the paper, which has been adjusted for age. It is a randomized trial, and we should expect the age to be equal in the 2 groups, but there was a slight imbalance, and so there was some adjustments. It's a very small adjustment, as you can see, 61.8% to 62.1%. And it's the statistician's job to carry out those more complex calculations. And details of those methods are all in the Lancet paper. You will also notice the 95% confidence interval, 41% to 76%. It's quite wide. And that is the range that describes the uncertainty about the estimate of efficacy. The true value of efficacy would be the value we'd get if we could test the whole population. Here we have a sample. The confidence in it refers to the probability that that confidence interval would contain the true population value when you select another random sample from the population many times. In other words, you repeat the experiment many times, but of course we don't do that. We work on one sample and we present the 95% confidence interval, and there's 95% chance the real value lies within that. I'm now going to look in a little bit more detail at the calculation. So here I'm going to do it in quite a simple way. We assume that each arm contains the same number of participants. It was randomized, it should have done, it was 1-to-1 randomization, and they are nearly equal. So we assume they contain the same amount, about 4,500. Now, we had 71 infections in the control arm, which wasn't protected with the vaccine. So the expected number of infections in the vaccine arm, if the vaccine is not effective, would be 71. But the actual number was 27. And so we calculate the percentage of reduction. And you can see I've put the calculation below. 71 minus 27 divided by 71, and then convert it to a percentage, and that's 62%. And so that is how we calculate the efficacy, and the efficacy that you see mentioned in the news and in the government's briefings time and time again from all the vaccine studies. Let's quickly have a look at that controversial group, the one that received the low dose as their first dose by accident. You can perhaps remember the controversy over this. It caused a lot of confusion in, well, everybody. And we didn't really get to the bottom of it until the paper was published, which has only been in January. And then we can see how it was calculated and where all the patients ended up in this very complex study. But here I'm just looking at the people who received the low dose, and you can see there was only about nearly 1,400 in each arm. And we had 30 infections in the control and 3 infections in the vaccine arm. Now that was, so we had 1/10 of the infections in the vaccine arm. And when we do our calculation for efficacy, we get 90%, which is much higher than the 2 standard doses arms were showing us. And that was where the confusion arrived, because they were reporting both efficacies. Let's just have another look in more detail, and you can see how I have calculated the 90%. So we would have expected 30 in the vaccine arm, but the actual number was 3. So the percentage reduction was 30 minus 3 divided by 30 times by 100, 90%. So that's how we ended up with a vaccine efficacy of 90%, but specifically for the patients who had appeared in the low dose, standard dose arms. Notice also, we still have a large confidence interval, 67.4 to 97, quite a range of possible values. Now, you've also asked about the dexamethasone study. And so I'm just going to show you briefly details of that study. This is published from in the New England Journal of Medicine, and I've extracted some slides that they published. It's the dexamethasone trial in hospitalized patients who've infected with COVID-19. And the trial was called the recovery trial. The recovery trial was testing drugs which may have been of benefit. This is known drugs that have already been tested in lots of other diseases and situations. They were being tested to see if they were in benefit in treating severely ill COVID-19 patients. Now, there was quite a few drugs tested and most of them showed no benefit. But dexamethasone, which is a steroid, and it's a man-made steroid, and it's used in lots of other inflammatory conditions like arthritis, asthma. It did show benefit. I'm going to now show you a brief description of the study. The patients are seriously ill with COVID-19. It's a controlled study, but it's open label. So it wasn't blinded. It was randomized. It was randomized in the ratio of 1:2. So twice as many people got usual care as had the dexamethasone, which was either orally or intravenous in a vein. It was given at the dose of 6 milligrams per day. And the primary outcome was mortality at 28 days after randomization to the study. Now, the investigators were interested in 3 subgroups. So the patients could be divided into one of these 3 subgroups at the time they were randomized. There were the patients who were receiving oxygen only, the patients undergoing invasive mechanical ventilation, and the patients receiving no supplemental oxygen. They wanted to look at the results separately for those 3 groups, as well as the patient group overall. Now, here are my summary of the results. The results are presented in the New England Journal paper as Kaplan-Meier failure curves for 28-day mortality. And I'm going to show you the graphs next in my next slide. And there's 4 graphs. Panel A is all patients, showing the numbers as for 28-day mortality. Panel B is those undergoing invasive mechanical ventilation. Panel C receiving oxygen without the invasive mechanical ventilation, and panel D receiving none at the time that they were randomized. So I hope you can see those graphs clearly. Let's have a look at panel A, and I will now try and answer your question of what is a Kaplan-Meier failure graph. So let's have a look at the axes on panel A. The horizontal axis is days since randomization. And remember that I said we were interested in mortality up to 28 days after randomization, which was when the patient started receiving the dexamethasone or the control treatment. The vertical axis is the percentage mortality. If we have a look at 28 days, we can see that roughly 26% of patients in the control arm have died, and in the dexamethasone, a bit less, about 23. The graph itself shows us how the deaths occur over the 28 days, and each step in the graph as it goes up represents more deaths occurring. So you can see they happened quite rapidly in the first 14 days, and then the rate slowed off. The numbers underneath show us how many patients we had to start with, and then how many we had at each 7 days throughout that trial. And then the other 3 graphs show us for the 3 separate groups of patients. And we can see that dexamethasone appears to have benefit in the patients receiving mechanical ventilation compared with usual care. It has benefit in those receiving oxygen only, but it doesn't appear to show any benefit for those who are not receiving oxygen. So there we have the four Kaplan-Meier curves showing us how patients fared through this 28-day study. I've now extracted some of those results so that we can have a closer look at them. And if you have a look at the bottom line, all patients, you can see that, notice that we have roughly twice as many patients in usual care as dexamethasone. That was because of the randomization ratio. We have a smaller percentage of patients dying in the dexamethasone group than we do in the usual care. And the ratio of those two, 22.9% divided by 25.7, is called the rate ratio. And it's about 0.9 for all patients. If we look at the three groups separately, it's the best rate ratio is for those receive an invasive mechanical ventilation. Oxygen only, it's 0.9, and then with no oxygen, it goes in the opposite direction where there is no obvious benefit of dexamethasone. Once again, I have to say that the rate ratio is adjusted for age in the published report, so the statistician would have carried out that more complex calculation. Now, this is a graph that is actually published in the New England Journal. And that is just a summary of the results I've just shown you, but in a graphical form. And you can see that we're looking at the rate ratios here, and those on the left show dexamethasone was better, and those on the right show usual care was better. Although, and one in the middle would show that there was no difference. We can see that those who received no oxygen, the line did actually cross one, so the confidence interval is quite wide for those patients. So there seems to be some clear benefit for dexamethasone, and they report an overall rate ratio of 0.83 and a p-value less than 0.001. Now, you also asked me, what does the p-value mean? Well, let's think about the dexamethasone trial and hypothesis testing. Now, most statistical analysis is based on hypothesis testing, and it involves some comparison between the treatment and a control for the outcome measure. And the numerical measure of that is called the effect. And the effect in the dexamethasone trial that we're interested in is the rate ratio. The null hypothesis is that the rate ratio equals 1. That means that there's no difference between dexamethasone and control, the usual care. But our result shows that the rate ratio is actually 0.83, with a confidence interval 0.75 to 0.93 and a p-value of 0.001, then it favors dexamethasone. That means that the probability, the p-value, 0.001, that we could have obtained the observed data or more extreme if the null hypothesis were true is very small. It is less than 0.001. So that means that we can be quite certain that dexamethasone shows benefit in these patients. Thank you all very much for listening. I'm afraid we've covered a lot of topics very, very quickly, but of course only briefly. So how can you learn more about statistics? Well, I say, invite the study statisticians to your meeting with the study investigators, and then the statistician can answer your questions directly. Us statisticians are very keen to meet patients, carers, and members of the public. We all have a common aim to improve the quality and relevance of medical research. Thank you all so much for listening.